right, good morning. Yes, so I'm Robin Gerlach, professor in chemical and biological engineering at the CBE. And I'm actually going to tie into a number of things that Matthew talked about, although on a much higher level, not on a molecular level. I'm preaching to the choir when I say that multi-domain biofilms are important. That's why you're here. And we find them in a number of environments. And my passion is probably in the environmental applications, but it really goes over into biotechnology, wastewater treatment, and more recently also into medicine, although I'm going to present some data from Garth James today on the multi-domain biofilms. Matthew already brought up this example of basically a bacterium archaeum co-culture, and he showed some images, so I'm not going to go into much detail. But what's interesting, and this was kind of described in this paper, is that there are models on how these organisms arrange with each other. And Matthew did a much better job and spent much more time on explaining how the interactions come upon. He also talked about cobalt methane, and actually cobalt methane is being produced through an interaction of bacteria and archaea. Here is actually an image, a confocal microscopy image of a bacterium attached to coal, which is associated with some methanogens. And there's an SEM image where you can see association of cocci with rod-shaped organisms. And if you look at how methane is being produced from complex carbon sources like coal, it's basically bacteria like Clostridia-like species, but also sulfate reducer, which can convert complex organic compounds into short-chain fatty acids, including acetate, as well as CO2 and hydrogen. And the archaea, the methanogenic archaea, then convert either the acetate or the hydrogen CO2, or actually methyl groups that might be associated with some of the degradation products, to methane. And these are interactions that we're beginning to think about, and we've looked at them from a community level and in the biofilms and how the biofilm interactions are important in this. That's something that we are looking into right now. Matthew also showed actually these images of some work that we've done. This is about an 8-foot diameter, what we call RABR, rotating algal biofilm reactor. And this is actually grown in wastewater. This is in the laboratory, but open to the environment. So we basically form multi-species, multi-kingdom biofilms. And we've used these small-scale reactors to actually study but the photosynthetic zones and the activity in those, oxygen consumption as an example, as well as pursued strategies to promote lipid accumulation in these biofilms. And it turns out that when we image them, first of all, they're beautiful. So here you can see the red is the autofluorescence from the chlorophyll, and the green is actually Nile red fluorescence, which appears when the cells accumulate lipids or other hydrophobic compounds. So on the top, you see actually the top of the biofilm of this mixed species, mixed community biofilm. On the bottom, you see actually the bottom of it, and you can see drastic differences here. So on the top, you have algal cells, and some of them accumulate lipids, and they do so. The right-hand side is in the absence of nitrate, so we let nitrogen run out. On the left, we have a resupplemented biofilm that does have ample nitrogen available. But on the bottom, you start seeing a very different community. And actually here, these blue strings, we believe, and I'll show you some evidence from other stains, are actually fungal cells that have established themselves. Bacteria we see every once in a while, but we really didn't see many bacteria, even though this was grown open to the environment in the lab. And then we use these biofilms and actually study oxygen consumption and production rates. So in the light, you can see, atmospheric, or based on atmospheric pressure, the solubility of oxygen is about 250 micromoles. You can see inside the biofilm, you accumulate oxygen, which then drops with the depth into the biofilm due to consumption, due to heterotrophs. And then towards the top of the biofilm, again, oxygen starts being transported outward. In the dark, you see consumption, net consumption again. Atmospheric equilibrium is roughly here. You see consumption, and one of the interesting aspects that we'd like to begin looking at is actually how the presence of other organisms in the biofilm potentially enhance the consumption of oxygen or reduce the net oxygen production during the light times. Now, this is an example that Garth James provided me with. There's obviously, you know, about yeasts that are associated with the body naturally, but that can become opportunistic pathogens. And Garth's lab has done some work on Candida and Malassezia, and there are a number of reactors that we're using at the CBE to grow all sorts of biofilms, and these were grown in CDC reactors as well as observed in these flow cells where we can insert coupons. And what you see is that depending on the interaction on the organisms, and I'm going to talk about inoculation and culturing conditions here in a little while on a different example, you see different arrangements. Here you see the Candida cells and some bacterial cells interdispersed, and here you see the Malassezia cells kind of on the top right, but also interdispersed, but it's a very different arrangement. Now at this point, I like to suggest that the scientific community really doesn't have a very good understanding or conceptual model on how these organisms arrange themselves. And Matthew did a wonderful job describing why that might be, because interactions that we thought we know between organisms are now beginning to be revised because we really don't know whether it's a mutually beneficial relationship, whether it's parasitic or similar. So there's lots of room to study this and lots of need to study it. What I want to do now is I want to slowly transition into an area where we are growing bacteria and fungi in co-cultures. We actually have also started working with algae, bacteria and fungi, but I'm going to focus on bacteria and fungi today. Now, before I go there, this is 2 biofilms. They're now, the scale was 200 micrometers, that were grown from wastewater, granular activated sludge, that are reducing selenium. And when the student Leah Tan came into my lab, she said, "Oh, I want to study these bacterial biofilms that reduce selenium." I said, "Well, you know, if they come from wastewater, you'll probably see a number of other organisms." And it turns out that depending on the conditions that she chooses, first of all, so the red ones are basically the ones that clearly reduce and precipitate selenium, in this case selenate. And some of them, once we started looking at it, so these are basically microscopy slides. They're about this long and about 1 inch wide, so 3 inch by 1 inch or 4 inch by 1 inch. And you can see that the biofilm morphology is very different. And some of these really looked like they were starting to accumulate some fungi. Now, it turned out that Erica Espinosa-Ortiz in my lab had studied selenium reduction by fungal biofilms. And she did a very nice job keeping everything out of there except for the fungi. So she grew pure culture fungal biofilms. You see these beautiful mycelia that formed. And in this case, again, this is a fungus, Phanerocate chrysosporium, that grows really thick biofilms. So this is almost a millimeter thick biofilm that accumulates this red substance, which is elemental selenium. So Erica then basically took bacteria and fungi and started growing those in different constellations. So for this study that I'm presenting, we used the drip flow reactor. Actually, if I go back, Erica used a modified drip flow reactor, which we ran horizontally. So these were completely saturated, these biofilms. Here, in this case, we actually have an angled drip flow reactor. Liquid is dripping in here, it's running down, so you kind of have a partially saturated biofilm that develops. And we monitored biofilm development over time, And again, we use Phanerococcus chrysosporium and Pseudomonas putida as examples. And this could be done with any of your organisms of interest. So we used the synthetic medium, initially grew at pH 4.5, inoculated with fungi and bacteria simultaneously to kind of establish feasibility. And then we watched this over about 4 days. Now, if we image this, we grew these biofilms on cellulose membranes. And going back to it, the next time we will not use cellulose because a lot of the organisms actually produce cellulosic compounds. If we stain it with chalcofluor, you see the cellulose membrane being stained. We can really not make out many fungi. Then over time, you can start the mycelium to grow, it forms hyphae, and ultimately we don't get enough penetration or we can cut out the cellulose membrane, so we now see the fungal biofilm. Using a different stain, FUN1, we again, kind of the dirty orange is the cellulose membrane, and we see a few cells, whether these are all fungal cells or whether there are some bacterial cells. If you look at the size, they're more likely to be fungal or at least elongated bacterial cells. We start seeing them to attach to these membranes, and then you can start again after 3 days to watch the mycelium form, and so you can get these beautiful structures. You can make out hyphae, lateral branching, you can see the separation between the different compartments, and so on and so forth. So we have the ability to image these, and we also have the ability to quantitate if we can reproducibly grow them based on image analysis. So then, this is really where the interesting part now comes in, looking at multi-kingdom, multi-species biofilms. We chose, again, the same organisms, Phanerococcus chrysosporium, Pseudomonas putida. We grew them at 2 different pH values, and we used different different treatments and different inoculation methods. So in one case, we grew them simultaneously. In another case, we pre-grew the fungus, then added the bacteria, then pre-grew a bacterial biofilm and added the fungus. And again, we did this at various pH values. So at pH 4.5, if we just grow a bacterial biofilm, we're able to establish it, and it doesn't look very thick, and it looks somewhat patchy. The fungus grows very well. If we inoculate the bacteria and the fungi simultaneously, and this is hard to see, but see, I think I can blow this up a little bit, you can start seeing a few green cells. And again, this is the live dead stain indicating that there are most likely bacterial cells there, but not very many. If we start with a bacterial biofilm and then add fungi, you see the fungi are kind of establishing themselves, but they have a harder time than in just a pure fungal culture biofilm, or if you grow them simultaneously. If you grow the fungus first and then add the bacteria, there are really very few bacteria to be found. And it's just the inoculation order can affect what you get out of the system in the end if you have these potentially competing species or organisms. So at pH 6.5, the bacterial biofilm, by and large, was much thicker, much more flush. The fungal biofilm alone also established itself. If we inoculated them, the fungus first and then the bacteria, we started not seeing a very thick biofilm in most cases. We're not quite sure why that is at this point. We haven't been able to do the bacteria and then fungus, so pre-grow a bacterial biofilm and then add the fungus. But in comparison now to the 4.5 system where when we added fungus and bacteria simultaneously, we see much more of a relationship between the fungus and the bacteria. And if I zoom into this one here, it actually looks like as if the bacteria are kind of more on the bottom of the biofilm and the fungus forms the mycelium up on top. Again, these are observations at this point in which we simply try to evaluate how different inoculation methods, different pH values, and so on and so forth affect biofilm structure. But it's interesting in itself that the order and the pH value of the cultivation medium really influences the outcome. So here's a clear comparison between the two. So again, this is simultaneous inoculation, pH 4.5, pH 6.5. So in the 4.5, the bacteria have a hard time establishing themselves in the biofilm, while at 6.5, they seem to be successfully either co-inhabitating or competing with the fungus. And again, this is one fungus, this is one bacterium, this is 2 pH values. So of course the matrix, the experimental matrix goes up, but I would love to hear suggestions on what to study, which organisms, and what type of inoculation methods and media to use. I'm going to skip, in the interest of time, over the pH values. There's not that much that we see. They all establish themselves, and in general, the pH goes down, presumably due to a reduction in oxygen availability and partial fermentation, production of organic acids from glucose as the principal carbon source. Now, interestingly, what I said about the biofilm with respect to the bacterial cells does not necessarily hold true about the cell counts in the effluent. In all cases, so all these systems that I showed you, we see microbes exiting the reactor. There are different concentrations, and again, remember there's a log scale, but the bacteria can be detected in the effluent even though we cannot easily detect them in the biofilm. So the bacteria do, over a period of at least 4 to 5 days, survive and maybe even propagate, but in some cases they cannot establish themselves in the biofilm. So we might be, in this case, which is counter to something that we often say as biofilm engineers, is it might be very informative to actually look at the effluent if you want to look at bacterial cell counts, because you can do this in selective media as opposed to using microscopy inside the biofilm, which might have a very limited detection limit, meaning high detection limit. Now, one aspect that we initially tried to not pursue a whole lot was reactor failure. So you drip medium into this reactor onto a slide, and then it kind of percolates down. And this is the point where the drop hits. In the fungal-only biofilm treatments at pH 5, we saw on several occasions that the fungi grew their mycelium all the way up to the needle that we have up here and basically plugged it. The biofilm, as a consequence, started drying out. And once we started looking at it, it looked like it was a very hydrophobic layer. And so we started going into the literature, and there is actually something that's called hydrophobins, which are being produced by fungi, and I have a slide on it here in a second. And there's actually industrial use for those potentially. But it basically happens when the fungal biofilms dehydrate, they start forming spores, and it's a hydrophobic, water-impermeable surface. And that is actually where some of the industrial applications come in, like surface coating. But before I get there, these are often cysteine-rich proteins. They have a high hydrophobicity, and people have suggested they could be involved or could be used for biosurfactant use as surface coatings, stabilizers for foams, and so on and so forth in drug development or drug delivery. So even though we set out to basically just study the interactions of bacteria and fungi and algae, but I didn't talk about that today, in biofilm systems, there are potentially industrial uses for this, and we are going to pursue this in some detail, actually trying to identify what these compounds are and how effectively we could produce them. So with that, I want to close up and thank the two postdoc and student, Leah and Erica, from Mexico and the Philippines, who stand right around, somebody from Pakistan in my group with a Chinese, as well as a Turkish student. So I love the diversity in my group, and I love the input that they have on the research, and I hope I'll be able to continue my research in the same manner that I have been. And with that, I'll try to entertain any questions.
I'll ask one question. Oh, well, she's getting the microphone. We had a discussion last night about the representativeness of the drip flow reactor and whether you saw consistent biofilms across the surface of that reactor when you're trying to grow a fungal bacterial biofilm. Now, you mentioned that you see this pillar grow up toward the needle, which drops the median. Do you feel like the rest of the biofilm is kind of compromised in terms of that representativeness, or do you think it can be an accurate model? I mean, I guess what I'm really getting at is how repeatable is this system for the fungal bacterial interactions that we want to study?
Yeah, so there are definitely gradients. And I guess there's always the question between how representative it is and how repeatable it is in time and space. So I can't tell you how repeatable it is between experiments yet because we've only done a few. There are definitely gradients, so what you see depends on where you sample. But that doesn't mean it's not representative of many environments where you do have water running down the side of a surface. So for standard testing, if you need to have the same coupon, say, that you might want to use for the same antimicrobial test, this might not be the right system to study the development of biofilms and the gradients that develop not only into the depth of the biofilm but also along a biofilm. I believe that this could be a good system. There was a question right there.
Yeah, and just following up on that question, you also were using my microscopy slides, and I was wondering if you could talk a little bit about the reproducibility of being able to understand the biofilm when you're just looking at such a small portion of it, or how are you doing that when you're...
Yeah, so in this case, we didn't set out to develop a repeatable standard method that we could immediately use for antimicrobial testing. This is to understand how bacteria and fungi, in this case, interact. We actually used membranes that we placed onto these coupons, and then we took the membranes off onto the glass slides and actually did microscopy. And yes, if you do microscopy, you're very limited in your field of view, and as I showed, you're also limited in your ability to detect certain community members like bacteria. We know there were bacteria in there, but they were hard to see. Now I can imagine a number of different reactor constellations if you want to go into testing of antimicrobials to grow more reproducible biofilms between coupons, but that's not what we set out to do there. And we can talk about it more. I think I answered your question. Okay. All right.
Okay. If you have any other questions, be sure to catch Robin during one of the breaks or today or tomorrow. Thanks, Robin.